I think everyone is here. So before we start, an announcement from the organizers that the poster boards are, you must have seen them in the area outside. And for poster presenters, they will have your name on it. So you should locate the board with your name and put the poster there. Okay, so let's start this session again. It's a pleasure to introduce Daniel Larson. He's from the National Cancer Institute, NIH, USA, and works on various aspects of gene regulation, starting from the dynamics of the macromolecules up to larger scale and computational methods and single molecule and so on. He's going to tell us about kinetic proofreading and gene regulation. Over to you.
Thank you very much. It's a pleasure to be here. It's a beautiful campus. I'd like to start with a motivation, the kinds of images we've been obsessed with for many years now. These are cancer cells, MCF7 cells in this case, and each one of those green dots is an RNA. It's going to be clear how we do that in a second. And when you look at these cells, you find some cells which have a lot of RNA, and they're adjacent to cells which have very few RNA. And this basic observation has set us and many others on a quest, which has now gone on many years. I think the questions that we're trying to answer are still very much the same as they were before, which is how much of this heterogeneity is genetic, how much of it is non-genetic, is it static or dynamic, and is it functional? And this field really started with imaging, but now it has evolved, shall we say, into single-cell RNA-seq. And so you can go to whole meetings now where all you see are these UMAP plots. So this is a dimensionality reduction, but it's still the same basic questions about variability in gene expression. And so historically, we have always punted on this question about what this means and what the consequences are. But we use this as a window into mechanisms, and specifically into the mechanisms of transcription. And our favorite approach for doing this is live cell imaging. And here I'm showing you these same MCF7 cells, but we've labeled the gene TFF1, which is a famous estrogen-responsive gene. It'll be familiar to many of you in this audience. We've labeled it with these MS2 hairpins, and we've inserted it into the endogenous locus. And so what you see in the nucleus is a polymerase which starts making RNA. It forms these hairpins. The hairpins bind a fluorescent protein. The polymerase continues down the body of the gene. It terminates, the RNA diffuses away. And so this is a tetraploid cell line. It has 4 copies of the gene. We've managed to label 3 of them. And so I'm showing you here these periods of on and off transcriptional activity, which we and others call transcriptional bursts. And these movies are 15-hour movies. So what I hope you take away from this is that this TFF1 gene actually transcribes quite infrequently during this 15-hour time series. Here's another view of that same data. So this is intensity on the y-axis. This is time on the x-axis. So this would be a single nucleus. You see the red allele bursting and also the green allele. You see instances where the red allele turns on and then is off for 12 hours. Meanwhile, the green allele continues to transcribe, and et cetera. And I'm going sort of briefly through this data because it's published. But the point being is that these very long off periods, which you can see here in this probability distribution, are really what drive heterogeneity at the single cell level. So sometimes these alleles just stochastically switch off for long periods of time. So labeling that gene was a true labor of love. The postdoc who did that work was on suicide watch. We started this work first with talins, which probably the younger members of the audience have never heard of. And we finished it with CRISPR, but it was laborious. So then we devised an approach based on an old fly genetics trick called a gene trap, where we now built a construct which has these stem loops in it, but it has these other bells and whistles. The most important one, we insert this splice acceptor, which is artificial. And so if this lentivirus integrates into an intron, the upstream exon thinks that this is a downstream exon. They fuse together, they splice together. And then we get resistance, and we can isolate clones and sequence to find out where they're at. You don't need to absorb all that. What you need to absorb is that using this approach, We labeled over 900 unique genes with MS2, and we labeled almost 9,000 unique genes with PP7. And so we can now do these bursting measurements really, I wouldn't say genome-wide, but over a fair number of genes. And so what you're seeing here, each row is a single gene. The white spots are where transcription is occurring. The dark spots are where there is no transcription. This is, again, 15 hours of single molecule imaging, but now in 96 well plates. And I hope when you look at this, you'll agree with me that transcriptional bursting is how all human genes are transcribed. Full stop, right? We have yet to find examples otherwise. And yes. Yes. Yeah. So I can summarize nearly 2 decades of work on transcriptional imaging by saying that the human nucleus is actually quite hostile to transcription. Transcription is a really rare event. It happens briefly, you make a few RNAs, and then the gene turns off. So here's some examples of data from that experiment. So this is the gene ERR-FI1. This is the glucocorticoid responsive gene, the gene row A, and this channel SLC2A1. And this just kind of gives you a sense of what this raw data looks like. Again, intensity on the y-axis, time on the x-axis. When we see these bursts, they're very similar from gene to gene, and they're very similar over time. Okay, so we want to understand, again, the molecular basis of this and why transcription works this way. We see this as a window into genes turning on and off just over and over again. And so we wanted to specifically understand how transacting factors, transcription factors, control this process. And so we went after maybe the mother of all transcription factors, which is the oncogene c-Myc. It's interesting as an oncogene because it's never mutated, so its oncogenic effects are mediated entirely just by overexpression. And around the time I was starting my lab, there were these reports that c-Myc, in fact, was a universal amplifier of transcription. It didn't execute gene-specific programs, but just turned transcription up across the genome. And so we thought we could use this as a model system, and the system that we devised was a photoactivatable MYC. And so this photoactivatable MYC is based on the love domain from plants. So this is how plants grow towards light. We didn't develop this. It was originally developed by Klaus Hahn. But what we can do is put this love domain so it shields this nuclear localization sequence. So when there's no blue light, the nuclear localization sequence is occluded in the love domain. We turn on the blue light, the nuclear localization sequence is exposed, and then MYC should rush into the nucleus. So that was the idea. This construct still transforms cells. So if you put it into NIH3T3 cells in a RAS G12V background, so this is our control, which is M cherry controlled by light. We don't see colonies. But then if we put in our PyMYC and we shine blue light on the dish, now we get transformation. So the construct is active. And this is what it looks like. So again, now the time scale is on the upper left. Whenever you see a blue box, that's when we turn on the blue light in the cells. So we turn on the blue light, and MYC comes in. It's very fast. It's reversible. It's a little leaky. I wouldn't say it's entirely tight. Excuse me. But with this construct, now we can do the experiment that I wanted to do, which is acutely perturbed transcription. In other words, we turn MYC on, MYC goes into the nucleus, and then we ask how the genes respond. And so that's what this experiment is. So here we turn the blue light on, MYC comes in, and we can specifically watch how these transcriptional bursts change in real time. So again, turn the light on, MYC goes in, and this is what we see. So if you look now at this burst frequency in a cumulative instead of a probability distribution, what you can see is that the on time of these transcriptional bursts increases in the presence of MYC, but this off time doesn't change at all. What that means is that MYC just takes every gene and tells it to transcribe just a few more polymerases, make a few more mRNAs during that on period, but it doesn't change how frequently the gene fires. So it's a very specific effect on transcription. And this was in contrast to, say, what enhancers do, which typically change the off time. Okay, so then we wanted to look at how proteins were responding, specifically in this assay. And for that, we're using a technique which Kazuhiro has already introduced, which is live cell imaging of transcription factors using the halo system. We do all this in microscope geometry, which is called halo. So this is, these are similar to Kazuhiro's images. So this is halo H2B. For us, it's a control as something that really doesn't turn over on chromatin on the timescales that we're looking at. But in contrast, if you look at a transcription factor, it's binding and unbinding chromatin and diffusing around the nucleus. And this is the part that we're interested in. So again, if you look at, this is now a chimograph. So this is time on the x-axis and position. And you can see these long binding events, which are characteristic of almost all histones in general. Linker histones might be an exception. Or in contrast, if you look at a transcription factor, it's coming on and off chromatin. Sometimes you see long events, but you also see this salt and pepper behavior, which is the dwell time on chromatin. And from data that looks like this and looks like this, we generate these survival curves. So this is the probability of a certain dwell time on chromatin versus that dwell time. So this is a log-log plot. And I'm going to show you a few of these, so it's important to understand what I'm showing you. Basically, what it says is that the most likely dwell time on chromatin is short. That makes sense. It's just a stochastic process. And occasionally, you get dwell times as long as maybe 100 seconds. But again, this is a log plot, so this is a small fraction of the events. So what happens when you turn MYC on? Well, so we can do this now for a variety of proteins. We can look at a sequence-specific activator, which is the glucocorticoid receptor. And we can look at these components of the initiation machinery, such as TVP, MED1, and SBT5. And we can look at it in cases where we have a control, which is pi-M cherry, and we can look at it in cases where we have pi-MYC. I know this is a fair amount of data. This is really more for the transcription aficionados, but I'm going to give you, this is really the lead-in to the part of the talk I want to focus on. The point being that when you turn on MYC, you have different effects on different components of the transcription machinery. I'm just going to show you two of them. One is MED1. This is the large subunit of mediator. And the other is TVP. This is the TATA binding protein. And what you can see is that MED1 is destabilized in the presence of MYC. So you see a shift of this black curve to the left, whereas TVP is stabilized on chromatin when MYC is in the nucleus. So it shows an increase. So I feel like I can summarize this work and probably work from dozens of other labs over the last 10 plus years and say that transcription factors turn over on chromatin at a very high rate. We can argue about the specific numbers, but these are rough estimates, which is that the non-specific dwell time on chromatin is around, let's say, 2 to 1 second, and the specific dwell time is around 30. So that means specific, when that sequence-specific activator binds its cognate site in the genome, it's typically bound for 30 seconds to maybe a minute. And this is a problem. And I hope if you remember one thing from this talk, it's that I feel like as a field, we don't have an adequate explanation for this phenomenon. And I'll tell you why it's a problem. So you probably learned in your molecular biology textbooks that there's some sequence of events to get the transcriptional activation. Don't get hung up on the details. There's probably some loops involved. There's things binding and unbinding chromatin. The point being you have sequence-specific activators, you have general transcription machinery, ultimately you get RNA. And we typically draw these things as sequential events at thermodynamic equilibrium with forward and backward rates. Now, the problem with this model is that, yes, you have sequence-specific activators, but you also have a whole sea of non-specific activators. And these non-specific activators are actually in vast abundance. So, and the problem, I think, can be stated quite simply, which is that if you have one sequence-specific activator, which binds for 100 seconds, and 100 non-specific factors bound for 1 second, from the perspective of the promoter sees non-specific occupancy as much as it sees specific occupancy. So I feel like at a place like ICTS, I'm just going to state that and you're going to buy it, but you can show that mathematically, and I think intuitively it makes sense. But in this case, the error rate then is determined by just this ratio of the non-specific to specific. This means that the most likely off period is 0, and you can think of promoters as fractional occupancy detectors. So for the rest of the talk, I really want you to think about this from the perspective of the promoter, not from the perspective of the transcription factor. Now, you can solve this problem by adding additional steps and making those steps energy dependent to arrive at what I'll describe as a kinetic proofreading pathway. And really, this sort of kinetic scheme has almost magical properties. Again, I feel like I shouldn't say that at ITCS, but we're just going to let it lie for now. And I'm going to talk about those properties in a second. And of course, I'm not the person who came up with kinetic proofreading. It was originally described by John Hopfield to explain templated addition, so adding the right nucleic acid by DNA polymerase, or adding the right amino acid by the ribosome. There's a formulation which I find to be a little bit better, which is from Jacques Nino, I think more relevant for what I'm going to talk about. And in a feat of what I can only call astonishing foresight, Dan Hirschlag, the biochemist at Stanford, predicted that we might someday run into this problem vis-a-vis gene regulation, vis-a-vis transcriptional activation. And what Dan predicted, not knowing anything about transcription factor dynamics, because no one did at that time, is that if transcription factors bound only transiently to chromatin, genes would have a hard time knowing when to fire, when their specific activator was bound versus their non-specific. So what do you need for kinetic proofreading? Well, you need a branched kinetic process. And by that, I mean this arrow right here. The branch, in this case, allows a fully assembled transcription complex to go back to unoccupied chromatin in a single step. So you can get back to the beginning through a different pathway than the way you came. Another way to say that is this is a cyclic process. So you need to consume energy. So this is a highly ATP-dependent process. So that means you're breaking detailed balance. You have some forward arrows without backward arrows, where they're heavily biased in that direction. And one of the consequences is that you have a trade-off between efficiency and accuracy. So this is the cost of proofreading, which is to say that you may go around this cycle many times, burning these complexes before you ever get a transcription event. Now, if you're willing to pay that price, there are a number of benefits. One is now that the error rate can be nonlinear in the off rate. In fact, it can go to some arbitrary power depending on how many steps you have. The most likely off period for a gene is greater than 0 seconds. You might call that a refractory period. And now you turn promoters away from being fractional occupancy detectors to being dwell time detectors. So I would like to argue that there is good evidence for this proofreading process in transcriptional dynamics. So here is the gene MYH9. This is one of the genes we labeled in our gene trap assay. So this is the probability distribution of off periods on the y-axis, and this is the time on the x-axis. So like many genes, the most likely off period is around, let's say, 30 minutes. We've always known that there was this peak in the distribution, but let's be honest, it's pretty noisy. You wouldn't really want to do much with this curve. This is the fit, but there's also the most noise at these short times. However, now that we can look at our polyclonal data, where we're averaging over hundreds of genes really, right? So this is, we'll be able to resolve these peaks much better. So now we have more confidence in these short time periods. We can see this clear peak distribution, and we can fit with this, which is theory, which is the proofreading theory. It looks pretty good on the probability distribution. If you now look at the cumulative distribution, it's, I think, more persuasive. So this is now the cumulative distribution. Again, time. This black line is the same data I'm showing you here, which is the off periods over hundreds of genes, and the green line is the proofreading fit. So what are the molecular implications of this model? One implication which is important is that the activation domain is essential for specificity. Why is that? Because the activation domain on that transcription factor is what's recruiting the downstream steps. So in this case, specificity is not merely read by that initial binding event, it's also read at other points in the complex. And the way you get a complex is with the activation domain. So I would predict that this activation domain is essential for specificity, not because it controls binding, but because it controls complex assembly. Second implication, that there is an ATP-dependent biochemical process for removing incorrect complexes. And this process is essential for specificity. So, and I would argue we know very little about what this molecular pathway would be. So this is the process that takes chromatin with a PIC or a non-specific complex and wipes it clean. It could be the proteasome, it could be chaperones, it could be chromatin remodelers. I personally find that to be the most attractive. But I think this is an unknown area right now in transcription. And then finally, there is a refractory period following a burst. And this is the speed limit for transcription. In other words, you can't burst faster than this because such a gene wouldn't have kinetic proofreading. And this speed limit is reflective of this specificity need. Okay. I want to end with a few more phenomenological conclusions. This is the take-home message. So I would argue that transcriptional bursting, what we see, is really a consequence of this proofreading process. So in other words, it's not like there is bursting and proofreading, but there is bursting because there is proofreading. So I'm going to give you a few conclusions. I think really they're all the same conclusion, just worded in 4 different ways, and I'll see which one resonates the best. One implication of this is that gene expression variability is the cost of gene expression specificity. In other words, the cell puts a higher premium on having the right gene respond to the right transcription factor than caring about the absolute number of RNA per cell. It would rather get the specificity problem right than get the abundance problem right. So in that sense, heterogeneity in mRNA abundance, the thing that we've been measuring for so many years, is preferable to heterogeneity in mRNA identity. And this ultimately limits the information capacity of mRNA distributions. So what do I mean by that? For example, we use single-cell RNA-seq. We use it in both perfunctory ways and more meaningful ways. But trying to describe RNA-seq distributions at the level of individual reads, I think, is a pretty useless activity. And the reason for that is I would argue that you could just binarize that RNA-seq distribution and have the same information content. In other words, you can characterize every gene as off or on, because from the perspective of the cell, it's only trying to control the identity and not the abundance of that RNA. And when we do that, when we binarize single-cell RNA-seq data, we almost get the same UMAP plots than when we use the analog data. And one prediction from this is that the most variable genes respond to the fewest transcription factors. So TFF1, the gene that I started with, the gene that Dimple works on also, which is one of the most variable genes in the cell, should respond to the fewest number of transcription factors. So the genes where there's the highest premium on specificity will be the most variable. And I'm going to skip that for now. Okay, so that was my conclusion, and I'm just going to leave this up here maybe during the Q&A. I don't have time to talk about it, but I bet there are people in this audience who will be able to solve this problem we have. We've recently developed a system to visualize enhancers and mRNA at the same time, at the same locus, the endogenous locus. So these are eRNAs and mRNAs. We see this very bizarre behavior where eRNAs are almost anti-correlated with mRNA, and the time between the enhancer activating and the promoter activating is quite long, 40 to 50 minutes. So just ponder that. And if you have an idea where that comes from, please tell me. And with that, I will stop and take questions.
Thank you very much, Daniel. I think there'll be lots of questions, but let me get ahead with a quick question of my own. Sorry. So what the cell cares about is not mRNA, but protein, right? So is there a mechanism that stabilizes the protein numbers despite fluctuations in mRNA numbers?
Certainly. I mean, there's obviously post-transcriptional control, and then proteostasis is important to that. I don't think it's not as easy to characterize protein variation. It's not as clean mathematically, but 100% there are mechanisms to do that, yeah.
Okay. I lost count of who raised their hands first. I think Kundan was first, and then I'll come to you.
Thanks for the really nice talk. So I was just curious to know that since many of these experiments are also done in cancer cells, and cancer cells, given their aneuploidy, both at the level of the genome and at the level of gene loci itself being higher in copy numbers. So I was just trying to think about how this might rationalize with the models that you present.
So yes, cancer cells are messed up. I will point out that the gene trap measurements that we do are not in transformed cells. They're in immortalized cells. Debate what that means, but they're mostly diploid. And they're CDK4 H-tert immortalized, but they don't grow on soft agar. They have contact inhibition, so it's a little better.
Azviru next.
So thank you for your nice talk. So with the increase in CMYC expression, so have you looked at the behavior of H2B hello?
No, we haven't. Should we?
Yes, I think so.
Okay, we will.
Shipra?
Yeah, so you know, there's a lot of work on variability and lineage specification, ESC differentiation. I don't know that work as well as I should, but certainly genes like NANOG and SOX2 seem to show increases in variability before lineage specification or during. And maybe somebody in the room knows this work better than I do. But you could argue that that is a moment in the cell's trajectory where specificity is highly important.
Sandeep over there.
That's just a speculation.
Very nice talk. So a couple of questions. The first one is, how much of the total energy budget of the cell, this amount of energy that you require for transcription, like what's the fraction, like what percentage of you that? And the other question is, say that I take a thermodynamic model, which is more complex, or say that I consider...
What kind of thermodynamic model?
Say with higher order interactions and so on. Okay. And there has been such proposals. I wonder how they compare to these, because it seems that those can also explain some of the things that you see. I don't know, I mean, that's what at least Jeremy Gunawardene and these people were suggesting. So can you discriminate between those?
I think maybe Jeremy's models are similar to ours, right? And these are sort of these negative reciprocity cyclic models. I'm not, they probably have proofreading properties, even if they're not called a proofreading pathway per se. And in terms of the energy consumption, it's a lot. I don't know what it is. I mean, I know if you deplete ATP, nuclear dynamics grind to a halt, things stop diffusing. It's fairly quick. So I don't know the exact balance.
Rahul?
Yeah, sorry, I can't see you, but go ahead.
So since I have the mic, I'll take the liberty. Thanks for the nice talk. You know, just to be a bit provocative, because you know, in any dynamic system, the idea of that you error correct yourself is what happens in biochemical systems. So I want to understand, what is the take-home message? Is the time scale the take-home message, or what should I take from this?
The take-home message, the take-home message first is that there's a problem, right? And I hope and I think a lot of molecular biologists haven't acknowledged that. So that's already a start. That transcription proofreading, kinetic proofreading can solve that problem, but the cost of solving that problem is gene expression variation. And maybe the cell is willing to tolerate that. And that I think there's a branch of transcriptional activation that we don't understand, which is the proofreading step.
Yeah, no, but the fact that you have an increased time scale, dynamic turnover in a system like this, multi-complex system, is something to be expected, right? In any system, of course. Classical biochemistry might have missed it, but if you think of really dynamics of processes, this is what is given. So is the time scale of 30 seconds that you see, is that critical for the kinetics of turnover of the chromatin remodeling enzymes, the transcription factors, cofactors, and so on, and then see what is the fittest there, and then
It does say that that there should be some dwell times on chromatin which are longer, but they are in a very small minority. They might be And we could try and find those, but some may last as long as minutes, but it might be 0.1% of the events. Maybe we're not seeing those. But I don't know, I guess, I don't know if there are other kinetics ways out of this specificity problem. Proofreading is the only one I know of. But if you're saying that's the only way, then I'm happy to hear that.
Yeah, but we can talk about coffee. But is there a correlation with the RNA that comes out? Is there a time scale correlation between this kinetic step of initiation?
Yes. So I mean, we can use that proofreading model to explain the dynamics that we see of RNA synthesis. But importantly, it also explains the dynamics of transcription factors on and off chromatin. So it explains both of those things, which I think is a new development.
Question?
So you have an underlying model, of course, for these kinetic proofreading steps. So given the average and the distributions, is it possible to infer something about the underlying model, or is there a multitude of models which will reproduce your data?
It is. It's weak. The inference power is weak. I mean, we can do this, but if other three-state models, even ones that detailed balance at equilibrium, will do almost as well. I mean, you see a slight preference in a Bayesian information criteria. But the experiment that hasn't been done, I think, is the specificity experiment. And so that part, I think, is the important part. And the experiment I would love to do, but I don't know how to do, is I want to know for every RNA in the cell, which transcription factor was responsible for transcribing that for a specific message. Like I want to know how many came from GR and how many came from ER and how many came for that one message. That's the specificity measurement, and I don't know how to do that.